So what are the different media required for the growth of the bacteria that we are going to see in this chapter. So now, first we will see what is culture. So this is a term given to the microorganisms that are cultivated in the lab for the purpose of identifying and studying them. So basically the culturing is nothing but the growth of the bacteria, and when the growth of the bacteria is happening, and then after that we have to identify them, that is which organisms are they, and we need to study them. So for studying and for identifying, it is very important that we should have a culture, and culture is nothing but the microorganisms when we are cultivating them. That is called as culture. So basically the culture is the collection of the microorganisms for studying them and for identifying them. Now we will see what is medium. It is a term given to the combination of ingredients that will support the growth and cultivation of microorganisms. So medium is nothing but a combination of the ingredients. That is, many ingredients are used together and those will support the growth and the cultivation of the microorganisms. So that is nothing but the medium. So what they do by providing all the essential nutrients required for the growth, that is for the multiplication, in order to cultivate these microorganisms in large number to study them. So medium is nothing but the combination of the ingredients we will use, and we will allow the microorganisms to grow in this medium, and this medium will provide all the necessary nutrients, ingredients, then growth factors which are required for the organisms for the multiplication, that is for their growth. That is nothing but the medium. The food material or substance required for growing microorganisms in vitro outside the body is called as culture medium. So culture medium is nothing but a food material or a substance which we give to the organisms in vitro. In vitro means outside the body, in the test tube maybe. So in that we give them the food material and allow the microorganisms to grow in that medium. Reasons for the bacterial culture. Why do we do bacterial culture? So the first is subsequent clinical diagnosis. If we think that the patient is suffering from any kind of a disease, now to know which disease does the patient has, for that we need to do the clinical diagnosis. And clinical diagnosis is nothing but finding out which organism it is. Now if we want to find out which organism it is, then we need the culture as well as we need the medium. So that is the first reason why we do the bacterial culture. Then second is studying its morphology and its identification. Now if you want to study its morphology, morphology is nothing but size, shape, structure, and also for its identification it is very important, because if you don't have media, if you don't have culture, then we will not be able to do these things. Then bacteria have to be cultured in order to obtain antigens for developing serological assays for vaccines. Now during vaccine productions, we need the antigens. Now to obtain those antigens, we need to culture the bacteria, because when we will culture the bacteria, we will get out the toxins from them and then we will inactivate them, and that we will use in the vaccine production. So for that we need the bacteria. Then the next is certain genetic studies and manipulation of the cells also need the bacteria to be cultured in vitro. Now suppose we need to do any genetic testing, like to find out that DNA composition or any mutation, or is there any manipulation in that gene. Now if we want to find out all these things, we first we need the bacteria, because if we will have the bacteria, then the bacteria will contain the DNA, and that DNA we will be able to study. So the first important thing is that we should have the bacteria. Then the next is culturing on solid media is another convenient way of separating bacteria in a mixture. Now suppose if we have a mixture of the bacteria and we want all the bacteria to get separated. Now how we will separate the bacteria from the group of bacteria? So with the help of the media. So if we will have the media, there are different kinds of media, which is one of them is differential media. So if we are using the differential media and when we pour the bacterial culture on the mixed culture, what will happen? They will differentiate the bacteria. So that will help us to differentiate between the different types of microorganisms. Now microbial culture, which are used for growing microorganisms such as bacteria and yeast. If you want to grow the bacteria and yeast, we use the microbial culture. Now we have seen what is microbial culture. Then the next is the most common growth medium for microorganisms are nutrient broth and agar phase. So these two are very famous and very basic ones. Nutrient broth, this is a somewhat liquid type medium at the initial stage, and the second is agar phase, that is somewhat solid phase. So these two are most commonly used growth media for the microorganisms. Then we have specialized mediums are sometimes required for the microorganisms and cell culture growth. Now we need a different specialized media also for some special type of cell cultures. So those are different. Now culture medium. It is used to grow the bacteria. It can also be used to enrich the number of bacteria. That is to grow more number of bacteria beneath this. Then select for certain bacteria and suppress others. Now suppose we have a mixture of bacteria and we know that there are which organisms are present in that. And if we want some bacteria to be there on our plate, and if we need the other kind of bacteria to get died, then that type we use some specialized medium. What happens in that specialized medium is it will allow the growth of one kind of bacteria and it will suppress the growth of another type of bacteria which we want to suppress. So differentiate among different kinds of bacteria. Then the next important thing is it will enable us to differentiate between different types of microorganisms like gram-positive, gram-negative. These are the different types of bacteria. So it will help us to differentiate between them as well. Now what is the need for culture medium? Why do we need culture media? What is its importance? So it is usually essential to obtain a culture by growing the microorganisms in an artificial medium. So now if you want to grow the microorganisms artificially, that is outside the bodies, for example, say. So if you want to grow them, we need to give them the food, right? If you want to live, even human beings need food for their life. So in the same way, the bacteria also need food for their life. So that food is nothing but their culture medium. So if you want the microorganism to grow, then it is very important for us to give them the culture medium. So that is the importance. Then the second is, if more than one species or type of organism are present in each requires to be carefully separated or isolated in a pure culture. So now if we need one type of species or we need a pure culture, so in that case culture medium is very important. Because see, if we have two or more microorganisms with us and from that we need only one microorganism for our study. So how we will remove remaining two, right? So for that there is some specialized medium also which will allow our interested microorganism to grow and the other two to die, but they will not grow. So there are these type of mediums present also. Then the basic requirements of the culture medium. What does the culture medium requires? Now there are different parameters which the culture medium has. So first is nutrients. So among nutrients, the first is energy source. So from where does the bacteria will derive its energy? Like we do eat our food and we derive energy from it. So in bacteria, what is their energy source? We will see that. So the first is phototrophs. Phototrophs from the name itself. Photo is nothing but the light. So the microorganisms which need light for their growth are called as phototrophs. So the example for that is Rhodospirillum rubrum. So this is the microorganisms which need light for its energy. Then the second is called as chemotrophs. So from the name itself we come to know chemo is nothing but chemicals. So they require chemical for their growth. So chemical energy basically they use. So the example of that microorganism is E. coli. That is Escherichia coli. Then the second is carbon source. They do need carbon source as well. So what is the carbon source for that? CO2, that is carbon dioxide, and other organic compounds as well. Then next thing here is nitrogen source. So nitrogen source they derive from the atmosphere or from the nitrate or nitrite. Then the second thing which they require for their growth or which we provide in the culture medium is mineral salts. So among mineral salts there is sulfates, phosphates, chlorides and carbonates of K and G and Ca. Also they need a very suitable pH of 7.2 to 7.4. So all these are the basic requirements for the bacteria to grow properly in the medium, and we need to ensure that we are providing all these factors in an appropriate amount. Now we will see the common ingredients of the media. Now we have seen that media is very important for the bacterial growth. Now for that media to get completed, there are certain factors which make up the media. So what are those? We are going to see in the slide. So first is water. Water is very important. So it acts as a vehicle. Pure or distilled water, all enzymatic controlled chemical reactions occur. Copper distilled water can't use. So water is like a vehicle. We all suppose we need to travel from one place to another. And if we are tired, we need a vehicle, right, to take us from one place to another. So in the same way, if the bacteria wants to grow in their life, so they need the water as a vehicle to move from one stage of life to another stage of life, like in the development. So it can be in a pure form or it can be used in a distilled water form also, so that all the enzymatic activities are in control. And copper distilled water we cannot use because copper will inhibit their growth. So keep that thing in mind. Then the second one is peptone. It acts as a buffer, hygroscopic in nature. It is hygroscopic, that is, it allows the media to absorb the moisture. Now we have seen in the previous lecture also that for the bacteria to grow properly, they require moisture also. So that moisture is given by peptone, because for growing, moisture is important. That's it. So the partially digested proteins obtained from meat, heart muscles, caseins, fibrin, soya milk, etc. So from where does this peptone been given to the media? From the meat, heart muscles, then casein, fibrin, soya milk, and also other sources are there. Then we have seen the previous slide, nitrogen source is also important, so that is also provided. Then protease, amino acids, inorganic salts, growth factors, nicotinic acid and riboflavin. So all these factors are additionally provided in the medium. Then the next is yeast, prepared from cells of Baker's yeast or Saccharomyces. So yeast is also provided in the medium for the growth of the yeast. They are providing the Saccharomyces and yeast for the bacterial growth, for different growth also. Yeast is nothing but it's a kind of a microorganism only. But if you want to grow bacteria, in that case, they need this yeast culture also. So it contains carbohydrates, amino acid, growth factors, and it can be used as a vitamin source. So this yeast which we see, the fungal type growth which we see on a bread or something. So, but obviously we will not take from there. We will take from lungs of its main source. But yeah, it is been acting as a vitamin source. Then the next is meat extract. It is fresh lean meat. So fresh lean meat is taken. It has been prepared. It is contains gelatin, protease, amino acids, creatinine, purine, mineral salts, carbohydrates, and vitamin B2. So all these ingredients are present in the medium. Now we will see the properties of agar. Now we have seen that for growing microorganisms there are plates of agar. So what is agar? Like we are making plates using agar. So what are its properties which helps the microorganisms to grow? For knowing that, we need to know the properties of the agar. So first is golden yellow granular powder. So it is a bit yellowish color, granular powder. There are granules of it. It is not very clean or smooth. It is granular like. Then used for preparing solid medium, that is solidifying agent. So if you want to make a solid medium, for solidifying, so it acts as a solidifying agent. It will give us a solid medium. Now nutrient broth, which we have seen, that is a liquid medium and this is a solid medium. Now for the medium to give solidifying effect, for that we are using this agar. Then obtained from different seaweeds algae. From where do we derive this agar? From seaweed algae. It has no nutritive value. It does not have any nutritious value. That is, it will not give any nutrition to the medium. Then not affected by the growth of the bacteria. Now when the bacteria is growing in the medium, the very good property of the agar is that because of the presence of the agar, the bacteria will not get affected. That is, its growth will not get affected. It will keep on growing. It will not cause any interference in the growing of the bacteria. Then resistance to action of all the microorganisms. When and whatever actions that the microorganism does, it acts as a resistance. So it will not allow any other activities to be carried out by the microorganisms. Then it melts at 95 to 98 degrees Celsius and remains liquid up to 40 to 42 degrees Celsius. So now when we are making nutrient broth, at that time we will keep it up to 40 to 42 degrees Celsius. So it will be present in a liquid form. And when we want to use it as a solid medium, at that time we will melt it till 95 to 98 degrees Celsius. So it gets solidified below 40 degrees Celsius. So below 40 degrees Celsius, it will get solidified. 2% agar is employed in solid medium. So now when we are making solid medium like agar plates, at that time 2% of agar is used. Easily available and economical. It is very easily available and it is economical. That is, it is not very costly also. It can be affordable. Now we will see the composition of nutrient agar. Now we are saying that it is a very common one. So what are its compositions? So first is, we can take yeast, meat, beef extract. Any one from this will work. We will take 10 gram of it. So what is its function? It acts as a source of carbohydrates, amino acid and other growth factors. So when we are providing any one from this, it will be acting as a source of carbohydrates, amino acids and other growth factors. Now the second is peptone. Now what is the role of the peptone? We have seen in the previous slide. It acts as a buffer. It gives a hygroscopic nature to the medium. Hygroscopic is nothing but it gives moisture to the medium. That we need 10 gram. Then it is a source of amino acid, nitrogen, sulfur, phosphorus and growth factors. So all this it provides. Then the next is sodium chloride. Amount is 5 gram. Its function is like electrolyte. Then agar, we will take 20 gram. It acts as a solidifying agent. Then we will take distilled water up to 1000 ml. And it is a vehicle. We have seen, like for the growth of the microorganism, water is very important. Like for human beings also, water is very important. In the same way, even bacteria require water and that acts as a vehicle. So this is the composition of the nutrient agar and this is very important and this is the basic and the common medium used for the growth of the bacteria. Now the classification. Based on physical state, there are 3 types of mediums: liquid medium, semi-solid medium and solid medium. Based on presence of molecular oxygen and reducing substance, whether we are providing molecular oxygen and the reducing substances, based on that there are aerobic medium and anaerobic. In aerobic medium, we will be providing them with the oxygen. In aerobic, sometimes sometimes no oxygen and the reducing substances. Then based on nutritional factors. Now what nutrients we are providing, which organisms we want to grow, what is the specialized organism that we want to grow from the mixture of the bacteria. Based on that, there are simple media, complex media, synthetic media and special media. Now we will see about the special media. Now in the category of special media, there is enriched media, enrichment media, selective media, differential media, indicator media, transport media and sugar media. Now first is enriched media and the second is enrichment media. So both means somewhat same. Enriching is nothing but if we have one organism and we just want to cultivate them, we just want to grow them for further analysis or for identifying them or for studying them. In that case, it is very important that we should have them with us in a good number. So for that we use enrichment medium. Then selective medium. Now when we have a combination, like when we have a population of different bacteria and from that we want to select only one organism. So for selecting that we will use selective medium, which will only allow our interested microorganism to grow in it and others will not grow. Then second is differential medium. Now when we want to differentiate between 2 organisms, that can be gram positive, gram negative. So in that case, we will use differential medium. Then the next is indicator medium. So it will indicate, like suppose if we think that if you have taken a patient sample and if we think that the patient is suffering from XX disease. Now in that XX disease, some kind of bacteria is present. Now if that bacteria is having a specialized activity, based on which we will be able to identify. So that will act as an indicator medium. Means in that medium they will do some specialized thing or their function. Based on that we will be able to identify them. In that case we will be using indicator medium. Then the next is transport medium. Now when we want to take microorganism from one place to another place. Now it is very important that they should be in a surviving phase. So for them we need transport medium so that we can travel. We can take them from one place to another place. Then the next is sugar medium. And that sugar medium, it's just a simple medium. The microorganisms which grows well in the sugar medium, we do provide them with the sugar medium and they will be growing in a proper fashion. The next is simple medium. Now, simple medium or the basic medium are culture media which contains the minimum adequate nutrition for non-fastidious organisms. So, the simple media is nothing but a very basic media. It will have a very minimum amount of the nutrition, and that minimum amount of nutrition will allow the growth of the microorganisms. Now, fastidious organisms only grow when specific nutrients are included in this diet. So fastidious organisms are nothing but those organisms which will only grow if we are providing them with the specific nutrients. If the specific nutrients are not there in the medium, they will not grow. So for example, nutrient broth or agar and peptone water. So these are very simple ones, but they do have a specific nutrient in them which will allow the microorganisms to grow. Then composition is lab length 10 gram, peptone 10 gram, NaCl 5 gram, distilled water 1000 ml. Now, when 2 to 3% agar is added, then we have it as a nutrient agar. So we have seen if we are adding 2 to 3% of the agar, then it acts as a nutrient agar, that is a solid medium. For semi-solid medium, agar concentration is 0.2 to 0.4%. Now when we want a nutrient broth type medium, that is a semi-solid, not fully solid, not fully liquid, semi-solid. So in that case, we are using 0.2 to 0.4% of agar. That is very minimum amount of agar we are using. What are its uses? It is basic of most of the medium used in the study at common pathogenic bacteria. Now, if you want to study, if you want to grow the pathogenic bacteria, then we do not need very high conduit media to be used for growing them. Simple media will work. So, and it is very, what we can say, it is not very expensive. It is affordable. So, mainly this agar medium is used for growing them, and it is used for subcultures of certain organisms. What is subculturing? Now, we have seen what is culturing. Culturing is nothing but growing of the microorganisms. Now, subculturing will be growing of the same organism or the same species from the original source. Now, suppose we have one tube containing microorganisms, full tube of microorganisms. Now, we need the same organism for many days for our study. So, what we will do, we will take one organism, we will strike them and we will grow them. That same organism, we will have another plate of it. So that is called a subculture, taking from main culture and growing it again. That is called a subculture. Then the next is liquid medium. Difficult to identify all types of organisms. When we are using liquid medium, it is very difficult to identify different types of organisms in them. It is suitable for isolation of bacteria from blood culturing and water analysis. When we are doing water analysis, when we are seeing whether the water is contaminated, when we say, right, the water has got contaminated, but how do we come to know that which organisms has contaminated the water? So in that case, liquid medium plays a very important role, and also blood culturing. For that also, liquid medium is more suitable than the other factors. The next is peptone water. It is basic liquid medium. So it is a liquid medium. It is not a solid medium. How is its appearance? It is clear. It is colorless. It does not have any color. It is watery. Then usually in test tube. It is usually found in test tube. We do keep them in test tube. What is its composition? Peptone. We take 10 gram. Then sodium chloride, that is NaCl, we take 5 gram. 1 liter. So this is the composition of the peptone water. So nutrient agar peptone water, these are very simple medias. These are used in day-to-day life in the culturing process to cultivate the pathogenic bacteria, and they are very important and they are very affordable ones. So they are widely used ones. So these are most preferred one. Then the next is nutrient broths. It is also used to test the formation of indole. Now suppose if any organism is forming the indole. So for that test also we use this nutrient broths. Culture of organisms for demonstration of motility. Now when we see the motility of the organisms, so in that case also we use nutrient broth. So this is a semi-solid type of liquid that or media that we are using. Then the next is complex medium. Complex medium have added ingredients for bringing out certain properties or providing special nutrients required for growth of the bacteria. Now complex media have many different ingredients added into them. For what? For bringing out certain properties. Like suppose if some bacteria is having some specialized category and that specialized function gets activated in presence of certain specialized ingredient, for example. So in that case what they do, they make a complex medium in such a way that they provide that special ingredient so that if that special organism is present, that will act on that special ingredient and we will come to know the presence of that special ingredient. So basically it is a combination of different ingredients, and they do provide certain additional properties also to the medium, and they provide special nutrients which are required for the growth of the bacteria. That is complex. From the name itself we come to know that it is a complex, that is it is nothing but a mixture of different different ingredients. Then the next one which we are going to see is synthetic media. These are prepared from pure chemicals and exact composition of medium is very well known. So now these are prepared from pure chemicals, like chemicals are used for making this media and the composition is exactly known because we are making this, right? This is synthetic. Synthetic itself tells us the medium that we prepared outside, right? So the composition is well known because we are making it, so we know how much composition we are using for it. The example is Gigo's medium. So in this, we know all the ingredients, how much quantity we are putting, so everything is well known in this medium. Then the next is semi-defined medium. In this media, the exact chemical composition of the constituents is not known because substance like medium peptone are used. Now when we are making semi-defined medium, in this the exact composition of all the ingredients are not known. Why? Because while preparing this media, we are using medium peptone. Now exactly we are taking in grams, that is right. But how much output it is giving, that exactly you cannot count. That's why they are saying that we do not know the exact composition of this. Now most of the culture medium used for routine diagnostic work are semi-defined culture medium. So diagnostically, the mediums which are being used, these are the category of semi-defined media. Like in hospitals also, when the clinical samples are being tested for the presence of different types of microorganisms, for testing them, the medium which they use very frequently are nothing but the semi-defined mediums. These are routinely used by them, and it gives a very good result for them, and it will work well in that category. Now we will see what is special medium. Now enriched medium comes under the special medium category. Now when basic medium is added with some nutrients such as blood, serum or egg, it is called as enriched medium. Now we come to know from the word itself. Enrichment is nothing but adding something new, adding something in addition, so that the bacteria grow in a very good condition. So when basal medium is added, in that we add some nutrients such as blood, serum or the egg, so that the specific organisms are able to grow in a proper way. When they are used to grow bacteria, which are more extinct in their nutritional needs. So the bacteria which need very good amount of nutrients, they do provide in this medium so that they are able to grow very nicely and very fastly. Then the example for these medias are Dawson's egg medium. It is a creamy colored, opaque slope kept in screw-capped bottle. So this kind of bottle is there. It is creamy, it is colored opaque, and it is a slopy one. It is a selective medium for isolation of Mycobacterium tuberculosis. Now when we want to isolate Mycobacterium tuberculosis, which is responsible for causing tuberculosis, so in that case we need a special medium, enriched medium. So this medium is being used for it. And composition of that is hens, egg and nutrient broth. So using these two, they make a media, and that media allows the growth of the Mycobacterium tuberculosis. So these come under the category of special medium. The next special medium is blood agar. Now this is also an enriched medium. Its appearance is red in color. Why? Because we are using blood. So when we are using blood, it will give color red. Then composition is sterile nutrient agar. We are using agar. Then dehydrated sheep blood. Whose blood we are using? Sheep blood. And what are its uses? It is routine culture. For the routine culture it is used. It is widely used in medical bacteriology. Then it also acts as an indicator medium showing the hemolytic properties of bacteria such as Streptococcus pyogenes. So if you want to find out whether there is a presence of Streptococcus pyogenes, in that case we use an indicator medium. Why? Because they show a special category, a special function, that is called as hemolytic activity. To find out that, we use the indicator medium, and that also comes under enrichment of a special medium category. Then the next is chocolate agar, also called as heated blood agar, because this blood is first heated and then it turns into chocolate color. That's why it is called as chocolate agar. But here also we are using blood. But why we are saying chocolate? Because after heating it gets chocolate color. That's why. It is a kind of a enriched medium only. Its appearance is chocolate brown color. When we heat that medium, its color gets changed to chocolate brown color. So the procedure is, the melt the desired amount of nutrient agar, cool it in a water, bath at 75 degrees Celsius. We take a nutrient agar, we heat it, then we cool it in the bath at 75 degrees Celsius. After that, we add 10 ml of sterile blood, that is which are free from microorganisms. We add that blood. We allow the medium to remain at 75 degrees Celsius. We allow the blood to remain at 75 degrees Celsius, that is we are storing it. Then mixing the blood and agar by gentle agitation from the time to time until the blood becomes chocolate brown and color within about 10 minutes. Then pour in place. So that blood and agar, we do a gentle agitation. Agitation is nothing but a slightly mixing of them. And for 10 minutes we do this, and after that we pour that in the plate. Now what is the use of this chocolate agar? Culture of Neisseria, Haemophilus influenza and pneumococcus. For growing these organisms, we need this chocolate agar medium. Then enrichment medium, it is a medium, it has a stimulating effect on the bacteria to be grown or inhibits its competitor. So enrichment medium, what it is doing, if you want to grow a specific kind of bacteria, it is allowing that bacteria to grow very nicely, and the bacteria which we do not want to grow, it is suppressing that organism. So the special category or the special property which the enrichment media has is, it will allow our interested organism to grow in a very good amount, and the organisms which we do not want to grow, it will suppress the growth of that organism. Now, how it is going to suppress the growth of that organism? We will have some kind of ingredients in our medium which will allow our interested organism to grow, and the organism which we do not want to grow, it will have some nutritory effect on that organism, and it will definitely not allow its growth. So, depending upon the composition of the medium, it will be decided. This result is an absolute increase in the number of wanted bacteria related to other bacteria. Just now I have said, the wanted bacteria will be grown, the non-wanted bacteria will not grow. Now example is selenite F broth. It is enrichment medium for culture of Salmonella typhi and Paratyphi bacilli from stool sample. Now from stool sample, if you want to isolate Salmonella typhi and Paratyphi bacilli, in that case we will use the selenite F broth. Now what is its principle? At neutral pH solution acid, salinity has high toxicity to coli from group of bacteria and not to most of the Salmonella group. So how is it working? At neutral pH, when we are giving 7 pH to the medium, what it is doing? It will not allow the coli forms of group organisms to grow, but it will allow the Salmonella group to grow. So based on the pH, it is decided that which organisms they are allowing to grow and which organisms they are not allowing to grow. If you are keeping neutral pH in the medium, then it will not allow the growth of the E. coli. Why? Because it is not suitable for them to grow at that pH. But it is suitable for Salmonella species to grow at that pH. So this is how this enrichment medium has been made. We are providing certain kind of a condition which is suitable for our intestinal organism, whereas it is not suitable for the other organism. And because of this, that organism will not grow and our intestinal organism will grow. So this is what is it mean. Then the next type is selective medium. Now it is a medium in which certain substances are present which inhibit all other bacteria except the desired bacteria. Now in selective media, it will allow a selective one bacteria growth, whereas it will inhibit the other bacteria's growth. That is a selective media. It is very selective. You say, right, selective means only one. So only one it will allow to grow, our interested one, and others it will not allow to grow. Then it encourages the growth of a particular species from a mixed inoculum. If we have a mixed culture bacteria and if we are putting onto the media, and that time we will select one kind of bacteria to grow, others will not grow. Why? Because it is a selective media, and by growing the microorganism on it, it is very selective. It will allow only one selective microorganism to grow on it. Now example is GCBS. It is a light green translucent medium kept in petri dish. So it is light green color. It is kept on a petri dish. It is a selective medium for Vibrio cholera. So for Vibrio cholera, we name this selective medium. Principle is bile salt inhibit the growth of normal commensals, that is unwanted bacteria. So on what principle it is working? It is working on the principle that it will, because of the presence of the bile salt, it will inhibit the growth of all other organisms and it will allow only the growth of Vibrio cholera, because bile salt is tolerated by the Vibrio cholera and not by other organisms. So this is how it will be able to grow Vibrio cholera. Now Vibrio cholera produces acid by fermenting of sucrose, which acts on bromothiamine blue indicator, producing yellow bodies. Now we are providing sucrose in this medium. So what the Vibrio cholera is doing, it is able to break down that sucrose, that is the sugar source, which acts on bromothiamine blue, which is indicator producing yellow bodies. So Vibrio cholera, what it will do, it will produce acid by breakdown of sucrose. That acid will go and act on bromothiamine blue, which is the indicator in the medium, and because of that the bacteria will turn yellow in color. So this is your selective media. The next category is differential media. In differential media, McConkey's agar is the famous one. Now McConkey's agar is a culture medium designated to grow gram-negative bacteria. It is a very useful medium for cultivation of Enterobacteriaceae. So McConkey's agar is mainly used for the growth of gram-negative bacteria, and also we can cultivate other Enterobacteriaceae also. Now it shows lactose fermenter as red colonies and while non-lactose fermenters as white or pale colonies. Now if the organism is producing lactose, we are giving lactose in that, and if the organism is producing acid, it will be able to ferment the lactose and it will give us red color. If the organism is non-lactose fermenting, then it will give us a white or pale colony. So based on doing that, we can differentiate between two different kinds of microorganisms. See this picture. In this picture, my monkeys are showing both lactose and non-lactose fermenting colonies. Lactose fermenting colonies are pink in color, whereas non-lactose fermenting colonies are the one which appears colorless. Now, in the whole medium, we are providing lactose, but now then, as we know, there are different types of bacteria present in us in that culture medium. Now, the organisms which are able to break down the lactose, that will get turned into pink color. The organisms which are not able to break down the lactose, they will remain colorless or yellow in color. So this is how we are differentiating between two different organisms, and this is shown on the slate. Gram-negative bacteria growing on the media are differentiated by their ability to ferment the sugar lactose. We have seen. Now, based on whether they are able to ferment the sugar or not, whether they are able to break down the sugar or not, based on that the gram-negative bacteria are differentiated. Now the lactose fermenter causes the pH to drop and is detected by neutral red. pH is below 6.8, which appears as bright pink to red colonies on the agar, and the one which are able to ferment the lactose. What is happening? The pH is getting low, drop over there, that is it is going below 6.8, and because of that the color is getting bright pink to red to the colonies. What are its uses? Acting as a visual pH indicator, the agar distinguishes those gram-negative bacteria that can ferment the sugar lactose, that is lac plus, from that those cannot, that is lac minus. The organisms which are able to ferment the lactose, that is lac plus, and the microorganisms which are not able to ferment the lactose sugars, those are designated as lac minus, that is they are not able to ferment them. Now this medium is also known as indicator medium, that is low selective medium also. So we can take this medium as an indicator medium as well as low selective medium also. Now actions of electrolyte serves to inhibit swarming by protease species. Now this will not allow the protease species to grow in it. Why? Because we are not providing electrolyte in that medium. If we would have provided electrolytes in the medium, it will allow the swarming of the protease species, that is gliding or the sliding of this protease species. If we are providing electrolytes, but we are not providing that in that. That's why it will not allow the growth of the protease species. Then lactose. By utilizing the lactose available in the medium, lactose bacteria such as, now the organisms which are able to ferment the lactose sugars are Escherichia coli, Enterobacter species and Klebsiella species. So these are the different species which are able to ferment the sugar, lactose sugar, and they are designated as lac plus. Will produce acid which lowers the pH of the agar below 6.8 and results in the appearance of red and pink colonies. How they are forming red and pink colonies? Because these organisms are able to produce acid. When they are producing the acid, they are causing the pH to get lower down to 6.8 or below that. And because of the lowering of the pH, the colonies are getting turned into red or pink color. The next is indicator medium. These media contain an indicator which changes color when bacteria grow on them. So the color of this medium basically gets changed when the bacteria starts growing in them. So this is the way, this is called as indicator medium. Then example is Wilson and Blair medium. So example of this medium is Wilson and Blair medium. For isolation of Salmonella typhi and Salmonella paratyphi, this indicator medium is used. They appear as black colonies. So black color colonies have been appearing on the medium. Now consider the black color of colonies is due to the ability of these organisms to reduce bismuth sulfide to sulfide in the presence of glucose coliforms are inhibited by VDL3 and bismuth sulfide. So how they are forming the black colonies? This is due to they are having the ability to reduce or to break down the bismuth sulfide to sulfide in the presence of glucose coliforms, and they are able to give us the black color. The next is transport medium. These are used for the temporary storage of the specimens being transported to the laboratory for cultivation. If we are taking the sample from the lab to the cultivation department, we need to transport them. We need the transport media to carry them from one place to another. So for this, we are using transport media. Now such media ideally maintain the viability of all organisms in the specimens without altering their concentration. So this medium will allow the cells to leave, but it will not alter their concentration. Means the concentration will remain the same and they will be able to survive in that condition. So that kind of conditions is provided in the transport medium. Then transport medium typically contain only buffers and salt. So this medium has only buffer and salt. The lack of carbon, nitrogen and organic growth factors prevents microbial multiplication. Now, in this transport medium, they are not providing carbon, nitrogen and organic growth factors, and because of this, the microorganism is not getting multiplied. What is our purpose? We just need the microorganisms to live alive. So, we don't want them to get multiplied. So, we are not providing all these factors. Now, transport medium used in the isolation of anaerobes must be free of molecular oxygen. Now, the transport media used for the isolation of anaerobes, it should not have the oxygen, because we all know anaerobes do not need oxygen. So, in the transport medium, we should not provide them with the oxygen. Then, lastly, we are going to see the characteristics of transport media. What are its characteristics? What are its properties? So, it should be non-toxic. It should not be toxic to the microorganisms. Then, it should not promote or inhibit the bacterial growth. It should not allow the bacteria to multiply, or it should not allow the microbacteria to get lesser in number. So we just want how many numbers of bacteria are there, we just want that much. We don't want to get lesser, we don't want to get multiplied also. Then it should be easy to carry and transport. It should be very easy for us to carry them and to transport them also. The examples of transport media are Venkatraman, Ramakrishnan medium, then buffered glycerol saline transport medium, and carry and Blair medium. So these all are the examples of the transport medium. So this all was about the culture medium.